So maybe I can start out by, a big thank you to Helen and Tom for setting this program up. I think it looks really terrific, and we're happy to be on board. Apologies for the somewhat bizarre cover art. This actually made much more sense on the last talk that I gave, and hopefully this will be a little more clear when we get to the end. About me, I joined the biology department here at BU a couple of years ago now. And what I'd like to do here is give you a couple of vignettes, or a very kind of soundbite look at some of the questions we're looking at in my lab, and how we think that some of these optical methods that you're going to hear a lot about today give us a little extra leverage on answering these questions. So I think I could describe my lab in a couple different ways, right? So I could tell you that we're a sensory neuroscience lab, and we're interested in how animals sort of take this wall of sensory information that they meet every day and sort of extract the different elements that are really useful for guiding their behavior. I could also tell you that we're sort of a learning and memory lab, because one of the things that we want to do with the sensory system is learn the specific cues that predict something useful about the environment, right? So this cue means something good is about to happen. This means something bad is about to happen. So how does this information sort of about these useful cues stored in the nervous system? So our window on these questions is the olfactory system. So smell, as you've probably heard, is really intimately linked with sort of all kinds of interesting behaviors in animals, to start with a simple example. So we've recently become interested in looking at pheromone processing circuits in the mouse, where we've got an example here. So we know that different sexes of the animal secrete different compounds that can elicit very, very different and relatively stereotyped behaviors. So here's a compound. If you present it to a male mouse, it tends to provoke fighting. Another compound presented to a male mouse, sorry for the PG-13 here, tends to provoke mating behavior. So we'd really like to sort of start getting a handle on some of the sort of underlying neural pathways that support these behaviors. So one approach that we're using is to use optical highlighters to try and outline or delineate these pathways and see where they send their information to deeper in the brain. So this is sort of a simple example where we're using a virus to express fluorescent proteins. This is in the olfactory bulb, and you can see where these guys send their axons sort of along the surface of olfactory cortex. So this is kind of a more of an old school anatomy approach. The wrinkle that we're adding to this technique is now we're using viruses that are actually conditional. They're dependent on previous activity. So what that means is here I'm showing you a set of neurons that are activated in the male mouse brain after mating with the female. Right? So out of this sort of complex soup of lots of different neurons that mean different things, these are the cells that are associated with this specific behavior. So we think that this gives us a nice foothold for asking, okay, where does the information from these neurons go downstream to different parts of the limbic system? And say here's, in contrast, is a different set of cells labeled by two males fighting with each other. So we're still sort of early on in this approach, but we think this will give us a nice way to sort of compare the anatomical basis, or where this information leads to in the brain that generates these very different behaviors. All right, so based on what I told you, you might get the picture of a mouse as sort of a little chemically controlled robot that just sort of encounters a smell and then executes some behavior, gets another smell, another behavior, and so on. But mice like ourselves are really sort of very social critters, and they spend a large part of their day interacting with other individuals. And they need to store the outcome of these encounters. So if mouse Bob has a big fight with mouse Stan, and Stan really puts the lumps to Bob, then he wants to remember this experience and sort of not avoid him in the future. Or actually the other paradigm that we're looking at is sort of mating is obviously another salient experience where animals might want to store some information about this. So we know that animals have a sort of a characteristic chemical fingerprint that identifies each individual. So we're using these optical highlighter approaches now. This is a transgenic line where we have expression of GFP under an activity-dependent promoter, and these are now in live brain slices. So what this lets us do is ask now functional questions about this optically identified set of neurons that we know is associated with this specific learning experience. So in this case, what we found actually is if we do intracellular recordings from these neurons, we know that the cells that are involved in the learning experience have a really strong loss of sensitivity, right? So this pheromone processing circuit now becomes sort of insensitive to the male with which this female is just mated. So using these tools to identify sort of interesting groups of neurons is a sort of a critical approach for us, but if you're like the reviewers of our papers and our grants, you might be saying, well, you should actually be sort of measuring what these cells are doing in the animals as they're awake and interacting and engaged in interesting behaviors. So we're making some strides in that direction with a lot of help from the Gardner lab. Thanks, Nathan. We are infecting the neurons in the accessory olfactory bulb with calcium indicators, and we're just at this point collecting bulk fluorescence with a fiber, and this lets us ask what the cells are doing when the animals are directly engaged in these social behaviors. So we're hoping to extend this to sort of these cellular resolution methods that I think you guys will hear much more about later on. All right, so why don't I end sort of our foray into pheromonal circuits here. And I guess the thing that really drew us to the olfactory system in the first place was sort of its really impressive ability to map almost any chemical stimulus onto sort of a good outcome or a bad outcome, or almost any infinite number of behavioral possibilities. So we know a lot at the initial stages of the olfactory system how information is encoded. So we've got sort of a huge number of sensory channels that respond to different chemicals. And if you spritz an odor on the nose of the mouse, this is imaging with CCD sensory maps in the olfactory bulb. And you can see that we get different maps for different odorants. So we knew a lot about the rules by which sensory information was encoded, but we knew sort of very little, these are much older experiments now, about how this information was sort of relayed to cortex and how cortex made sense of it. So in this case, we didn't really want to even just watch the activity of the neurons. We wanted to be able to directly manipulate the activity of the cells and use that to sort of probe the structure of the system. So what we did was used a photochemical reaction, where I think you can see here we're shining a UV laser on the surface of the olfactory bulb, and we're activating these neurons by releasing neurotransmitter at these different locations. So what this lets us do is activate these specific sets of neurons in a very systematic way, and ask, if we record from a neuron in the cortex, then which subset of these channels are these cells listening to? Right? So we have a nice way, in this case, to sort of map out in the intact brain what are the patterns of connectivity that get information from one part of the olfactory system to another. So I think I'm almost out of time here. Why don't I stop there? I'll just leave you with the idea that, sort of unlike Jerome, we're freeloading on the hard work of people like him who have developed these really great tools, and we're using these more from a biological perspective to understand sort of the structure of the nervous system and how it changes with some of these interesting learning paradigms. So from there, I have a 2-year-old who's sort of not very patient and waiting for a pickup, so I'm going to have to run, so I won't be able to chat with you guys tonight, but if you want to get in touch and talk any further, please get in touch, or I'm sure Helen can get us in touch as well. So thanks a lot. I'd be happy to take a question or two if there are.